It's my pleasure to introduce our next speaker, Margie McGlynn. Margie is the HCU Network America co-founder and board president. She will provide a treatment overview of classical homocystinuria, which will serve as a scientific foundation for this afternoon's discussion. Margie, over to you.
Well, thank you for that introduction, Danae. I became involved in homocystinuria as a young child. In the mid-60s, I had two sisters diagnosed with classical HCU. One passed away at age 14 of a stroke, and one at age 9 of a pulmonary embolism. I vowed that someday I'd do something about it. So after a long career in the pharmaceutical industry, I started a research fund in their honor, and I co-founded HCU Network America with Danae. So I'm now pleased to give you an update of how far the research pipeline has come, but I will first set up the current treatment overview. So I'm going to take you through the current treatments for classical homocystinuria. First, about half of patients, as you heard this morning, have a mutation that's responsive to vitamin B6 or pyridoxine, which is a cofactor. So those patients are obviously given that treatment because it can lead to a significant reduction in homocysteine levels. But the mainstay of treatment is really dietary approaches. Patients are prescribed a low protein or in some cases a low methionine diet. And the intent there is to decrease levels of methionine so it cannot be converted to homocysteine, which I refer to as HCY, and methionine is referred to as MET. But these patients also then need to receive a methionine-free amino acid supplement to support their growth. About 3/4 of patients are also given betaine to donate a methyl group to convert homocysteine back to methionine. But many of these same patients are also on dietary approaches and or pyridoxine. Patients often get supplements of folic acid and vitamin B12. Both are important for the remethylation pathway. And if patients have low cysteine levels, they will also receive supplements for that because that compound is not made if the CBS enzyme is defective. But despite all of these therapies, homocysteine is often not controlled. In fact, the natural history study results were recently presented at the SSIEM meeting that showed that the average homocysteine level of patients in established metabolic centers was 112 micromoles per liter. And that's compared to the upper limit of normal of 15. And 45% of patients in this study had levels over 100, putting them at higher risk for thromboembolic events. So we need better treatment. In fact, there have been no new therapies introduced since 1996 when betaine was brought to the market. And since my sisters were diagnosed in 1964, betaine is the only treatment that has come onto the market. And none of these treatments are disease modifying, and there are many issues with compliance and palatability. But there is hope for the future. Let me now take you through the pipeline, but first I'm going to take you back to the metabolic pathway that Dr. Chapman took you through this morning. I won't take you through it again, but I'm putting this up here because I'm going to come back and show you where all of the approaches that are in research affect this pathway. So there are 5 novel approaches being studied for classical homocystinuria, at least that we are aware of. The first is systemic enzyme replacement therapy, and this would replace the CBS enzyme so that you can degrade homocysteine. The second would be oral therapy that can prevent methionine absorption so it's not converted in the body to homocysteine. The third approach would be gene therapy to deliver the DNA to liver cells so that they can produce the CBS enzyme, which then degrades homocysteine. The fourth approach, chaperone therapy, to stabilize the CBS enzyme to restore function. And lastly, to modify the metabolic pathway in a way that can help lower homocysteine levels. So let me now take you through each of these. But first, I'll show you the pathway and where each of these approaches are targeted at the pathway. So you can see here on the upper left, preventing methionine absorption here, so it's now converted to homocysteine. You can see the metabolic pathway modification where formate can also contribute to this conversion. And then these three approaches are all targeted at the CBS enzyme, either replacing it or enhancing its stability so that it can function better. So now let me take you through enzyme replacement therapy. There is one product in development called pegtibatinase, which is a pegylated version of the CBS enzyme. This was discovered by the late Jan Kraus at University of Colorado. This program has completed phase 1/2A, and recent data presented on the highest dose cohort showed up to a 67% reduction in homocysteine levels. Travere, the company developing this therapy, has announced that they hope to initiate their phase 3 trial in fourth quarter 2023. The second approach, there are two products I'll talk about. Both are oral therapies to prevent the absorption of methionine so that you cannot convert methionine to homocysteine. The first is being developed by Synlogic, and it's an engineered probiotic bacteria that consumes methionine in the GI tract. This program has completed phase 1, where they have achieved proof of mechanism in healthy volunteers. They do have modeling data that suggests it could lower plasma homocysteine by up to 58%, but obviously that has to be shown in patients with classical HCU. The company has not yet announced their timeline for beginning a phase 2 trial. The second product in this category is from Petri Bio, and it's a bacterially derived methionase, an enzyme which degrades methionine in the gut so that it's not converted to homocysteine. This program has achieved preclinical proof of concept where they showed a 35% reduction of homocysteine in a mouse model. The next step would be IND-enabling studies so they can file the IND and begin the first phase of human testing. The next category, gene therapy, only has one active program, and that is an AAV vector that contains the DNA, which can then be transcribed to the CBS enzyme. It's a joint program of Warren Kruger and Ron Crystal. This program has achieved preclinical proof of concept in a mouse model, and they are now determining the best path forward for IND-enabling studies and moving on to phase 1, which will likely require an industry partner. The next category, chaperones, has one program currently active, although there have been other products in this category where data has been published in the past, preclinical data. The active program is at Newcastle University, Dr. Thomas McCorvey. This is at discovery stage where they are screening for molecules that could bind to the CBS enzyme and restore its function. The next step would be preclinical proof of concept. The last category, metabolic pathway modification. The only active program is formate program being studied by Ken McLean at the University of Colorado. Dr. McLean has demonstrated preclinical proof of concept in a mouse model, first showing that low-dose formate can lower homocysteine levels, and also showing that when you combine formate with betaine, you can achieve near normal levels of homocysteine in the mouse model. The next step would be IND-enabling studies and determining the appropriate dosage form. So this slide now shows you where each of these specific products affects the pathway. In the upper left, Synlogic and Petri Bio. Then you have formate, and then the enzyme replacement therapy, pegtibatinase, gene therapy, chaperone therapy, all addressing the CBS enzyme. So the last topic I'll cover are what are the issues that arise in the development of new therapies for classical homocystinuria. And I did ask the researchers for all of the programs I discussed to provide input on this. So this also reflects their thoughts. First, it is not yet confirmed what specific biochemical changes cause what specific clinical issues in patients. Certainly, it's been shown a correlation between overall homocysteine levels and thromboembolic effects, but what exactly is causing cognitive deficit, ophthalmic issues, etc., still is not confirmed. Second, there could be an issue of availability of homocysteine for enzyme therapy to exert its effect because homocysteine forms disulfides, so therefore the enzyme would not be able to clear it. On the other hand, the initial data we've seen would give us reason to believe that this may not be a problem. Probably the biggest problem is the difficulty of controlling diet during a clinical trial. While you can give patients instructions as to exactly what they should do, patient compliance with dietary therapy for HCU is very poor, and high variability could exist day to day. The next point, some of the researchers, particularly at an early stage, are looking for greater clarity on what is the appropriate endpoint and the development path to get to market. And if the endpoint is levels of homocysteine, what is the right target? Is it 100? Is it 50? And I'm sure as the earlier programs move through development, that will become clearer. And the last issue is there may be enrollment challenges for clinical trials for classical HCU because of the small population versus other diseases and the proximity of many of the patients to metabolic clinics. This can be overcome by things like remote visits being allowed in clinical trials. So in summary, the current treatment landscape leads to very poor compliance and lack of control of homocysteine. And as you'll hear about from patients, there are many quality of life issues that result from this. But there is hope for the future. Many new treatments in development that could possibly offer greater control of homocysteine levels, potentially even allowing relaxation or no dietary treatment, which is the main goal of patients. They would love to be able to avoid the low protein diet and the poor tasting supplements that come along with that. So we are excited for the future, and I'm pleased to have been able to share this overview with you today.